Hello there, and welcome to Sleepy Science Stories. Tonight, we are not going to outer space. We are not going to the stars or the black holes or the vast silence between galaxies. Tonight, we are going down. Down through the surface of the ocean. Down through the warm upper layers where sunlight still reaches. Down into the quiet, dim world below, where the water moves in slow and ancient rhythms, where small creatures drift like living lanterns, and where one of the most astonishing things in all of biology has been happening, quietly and without any audience, for hundreds of millions of years. The ocean is older than anything on land. Life began here. The first cells that ever existed on this planet were ocean cells. The first experiments in DNA, in protein, in the chemistry that would eventually become every living thing you have ever seen. Those experiments were conducted here, in the sea, long before there was a single green thing growing on any piece of dry ground. And because the ocean is so old, and because life has had so long to work with it, the ocean contains solutions to problems that the rest of biology has never needed to solve. Strange solutions. Quiet solutions. Solutions encoded in the genes of animals so small, you could miss them entirely if you were not looking for them. Tonight, we are looking for one of those animals. It is a jellyfish. A very small one. Smaller than your smallest fingernail. Its body is nearly transparent, and you can see right through its bell to the bright red stomach at its center. Around the edge of its bell, it carries between 80 and 90 white tentacles, each one fine as a thread, trailing in the water as it pulses slowly through the current. Its scientific name is Turritopsis dohrnii. You can say it like this: Turritopsis dohrnii, but most people who talk about it use a simpler name. They call it the immortal jellyfish. That is not a poetic name. It is not a marketing name invented to make it sound more interesting than it is. It is a name that reflects something genuinely extraordinary about what this animal can do when it is damaged, or starved, or old. It can reverse its own development. When this jellyfish is under enough stress, it does not die. Instead, it pulls its tentacles inward. Its body contracts. It sinks to the seafloor. And then, over the course of about a day and a half, it transforms. Its cells change their identities. Its entire body reorganizes. And it becomes a polyp, an earlier stage of its own life cycle, beginning again from a younger starting point. The biological clock, in a real and measurable sense, runs backward. This is not something most animals can do. In fact, it is not something any other animal has been confirmed to do in quite this way. The process is called transdifferentiation, and we will spend tonight understanding what it is, how it works, what the science says about it, and what it might eventually mean for our understanding of aging and life. We will take it slowly. There is no hurry tonight. The jellyfish drifts and pulses on its own schedule, and we can follow at a pace that feels easy and restful. If you enjoy these sleepy science journeys, please consider liking or subscribing so that other curious minds can find their way here too. Every listener helps this little channel reach someone else who might fall asleep tonight to something wonderful. But for now, let your body settle. Let your breathing slow. Let the weight of the day dissolve, and let your mind drift gently downward, toward the ocean, toward the water, toward a small transparent creature pulsing quietly in the dark. Before we follow this jellyfish into the details of its biology, it helps to sit for a moment with what kind of discovery it represents. Science makes two kinds of discoveries. The first kind fills in a gap in something already understood. It adds precision, or detail, or confirmation. The second kind changes the frame entirely. It reveals that something assumed to be universal is not universal. That a rule taken to be absolute has an exception, and that the exception, once found, changes what is imaginable. The immortal jellyfish is the second kind of discovery. For a long time, the progressive aging and death of complex organisms was treated as a biological inevitability, not a design choice but a constraint. something built into the nature of cells and chemistry that could not be fundamentally circumvented. And then, in the Mediterranean Sea, researchers working with a small, fragile, transparent sea antarian found an animal that, when damaged or starved or simply old enough, folds its body inward, dissolves its adult form, and rebuilds itself as a younger stage. An animal that does not simply resist aging, but reverses it. That discovery did not appear in popular news with the attention it deserved when it was first documented. It took years for the broader scientific community to accept it fully, and it is still being worked out, slowly and carefully, by a small number of researchers around the world who have committed years of their careers to understanding how a 4.5-millimeter jellyfish does something that the rest of the animal kingdom has not managed to achieve. Tonight, we will follow that story. Slowly. With the kind of attention that only a resting mind can give. The ocean is a good place to bring a resting mind. It has its own rhythms, slow and ancient, that are compatible with the pace of falling asleep. The sound of water. The absence of edges, the quality of darkness in deep water, where light fades gradually rather than stopping abruptly. If you have ever fallen asleep near the ocean, you know what it does to the nervous system. It slows things down. It makes the usual urgency of thought feel less urgent. It opens space. So let your body become heavy now. Let your shoulders drop, and let your breathing slow to something easy and natural. There is nothing you need to track or remember tonight. The science will unfold at its own pace, and all you need to do is let it move through you like water through a slow current, carrying whatever settles and releasing the rest. The jellyfish is already out there. It does not know you are coming. It pulses and drifts and carries on, as it has for hundreds of millions of years, without any awareness of the attention it has received. It is simply doing what its cells know how to do, and tonight, we will watch. Let us begin. Before we can understand what the immortal jellyfish does differently, we need to understand what it is escaping from, and that means understanding aging, what it actually is, where it comes from, why it happens to almost everything that lives. Most people think of aging as simply getting older, and of course, in one sense, that is exactly right. But biologically, aging is something more specific. It is the progressive accumulation of damage inside the cells of your body, combined with a gradual decline in your body's ability to find and fix that damage. Aging is not a single event. It is not one thing going wrong. It is many small things, happening slowly and simultaneously, adding up over years and decades into a pattern of decline that eventually the system cannot compensate for. To understand this, you have to start with the cell, because almost every theory of aging, every serious scientific framework for understanding why organisms deteriorate over time, is ultimately a story about what happens inside individual cells. A cell is a machine, a very small, very complex machine, but a machine in the truest sense. It has moving parts. It has fuel systems and waste disposal systems and communication systems and replication systems. It produces specific proteins for specific purposes. It maintains a barrier between its interior and its environment. It responds to signals from neighboring cells and from the bloodstream. It divides when instructed to and stops dividing when instructed to stop. And like any machine that runs continuously for a long time, it accumulates wear. Some of the most important wear happens to the DNA itself. Every time a cell divides and copies its DNA, there is a chance of error. The copying machinery is extraordinarily accurate, but not perfectly accurate. Over billions of cell divisions across a lifetime, some errors accumulate. Some of those errors affect genes that regulate cell behavior. Some affect genes involved in cancer prevention. Some affect genes involved in repair processes themselves, which is particularly unfortunate because a damaged repair system becomes less able to catch future damage. There is also a specific and well-studied mechanism involving structures called telomeres. Telomeres are protective sequences of DNA at the very ends of each chromosome. You can think of them as the plastic tips at the ends of a shoelace. They do not carry genetic instructions. Their job is to protect the functional parts of the chromosome from degradation and from being mistaken for a broken strand that needs repair. Every time a cell divides, the telomeres get a little shorter. This happens because of a limitation in the machinery that copies DNA. The copying enzyme cannot fully replicate the very end of a linear DNA strand, so a small piece is lost with each division. Over many divisions, the telomeres shorten to a critical length. When they become too short, the cell detects the shortened ends as damage and triggers a response. The cell stops dividing. It enters a state called cellular senescence. A senescent cell is not a dead cell. It continues to carry out many of its functions, but it can no longer divide, and it begins to release a set of inflammatory signaling molecules into its surroundings. These molecules affect neighboring cells. They can disrupt the function of surrounding tissue. And as more and more cells enter senescence over the course of an organism's life, the tissues they make up become less functional. They repair more slowly. They respond less effectively to injury and stress. The signs we associate with aging begin to appear. Telomere shortening is one mechanism of aging. There are others. Epigenetic marks, which are chemical modifications to the proteins around which DNA is wrapped, control which genes are active in each cell. These marks determine whether a given cell is a muscle cell, or a liver cell, or a nerve cell. Over time, those marks can shift and drift, losing their precision. Cells can become less confidently themselves. The molecular definition of each cell type becomes fuzzier, and the coordinated function of tissue suffers. Mitochondria, the organelles that generate energy inside each cell, also deteriorate over time. They have their own small genome, separate from the main DNA in the nucleus. That mitochondrial DNA is particularly vulnerable to damage because it sits close to the machinery of energy production, which releases reactive molecules as a byproduct. As mitochondrial DNA accumulates mutations, the mitochondria become less efficient. Cells with compromised mitochondria cannot sustain the level of activity that younger cells can. Damaged proteins accumulate too. The cell has systems for identifying and disposing of misfolded or damaged proteins, but those systems become less efficient with age. Protein aggregates begin to build up in cells, interfering with normal function. All of these processes, DNA damage, telomere shortening, epigenetic drift, mitochondrial decline, protein accumulation, they do not act in isolation. They interact with each other. They compound each other. A cell with shorter telomeres may also have less efficient DNA repair. A cell with damaged mitochondria may have less energy available for quality control. The different mechanisms of aging are not independent problems. They are interconnected threads in a single fabric of decline. Why has evolution not solved this? Why, after billions of years of natural selection, does almost every complex animal still age and die? The answer is subtle. Natural selection favors traits that improve reproductive success. An organism that survives long enough to reproduce passes its genes to the next generation. Selection pressure is strongest early in life, when reproduction is happening. Once an organism has reproduced, selection has much less influence over what happens next. Genes that cause problems late in life are not strongly selected against if those same genes are neutral or beneficial during reproductive years. The engineering incentive to maintain the body indefinitely was never strong enough to produce a universal solution. Almost never. The immortal jellyfish found a different path. Not because evolution was kind to it, but because, in its particular environment, with its particular biology, a different arrangement of molecular tools produced a different outcome. Let that thought settle gently as your breathing slows. Aging is not punishment. It is not failure. It is the natural consequence of being a machine that runs without perfect maintenance for a long time. And somewhere in the ocean, a very small machine has found a way to occasionally reset itself entirely. To understand what makes Turritopsis dohrnii remarkable, you need to first understand what an ordinary jellyfish life looks like, because the immortal jellyfish begins exactly like any other member of its group. It is only what can happen later that sets it apart. Jellyfish belong to a group of animals called cyanidarians. This group also includes corals, sea anemones, and hydras. Cyanidarians are among the oldest animal lineages on Earth. Their ancestors were present in the ocean more than 500 million years ago. They predate fish by hundreds of millions of years. They predate insects, amphibians, reptiles, birds, and mammals. They were here before the continents were arranged as they are today, and in all that time, through every mass extinction and every dramatic shift in ocean chemistry and climate, the cyanidarians have persisted. Their body plan has proven remarkably resilient across deep time. The defining feature of cyanidarians is their relative simplicity compared to most other animals. They do not have a brain in the sense that vertebrates do. They do not have a dedicated respiratory system. They do not have a heart or a closed circulatory system. What they have is a nerve net, a diffuse mesh of interconnected nerve cells spread across the body that coordinates movement and response without any single control center. They have stinging cells called cyanesites, which give the group its name and which are unique to this lineage. And they have a body architecture that, while simple, is extraordinarily flexible in how it can be organized and reorganized. The group of cyanidarians that Turritopsis dohrnii belongs to is called hydrozoans, and hydrozoans have a life cycle that alternates between two very different body forms. The first form is called the polyp. A polyp is a small, stationary structure. It attaches to a hard surface on the seafloor or on a rock. It looks something like a tiny tube or vase, with a mouth at the top surrounded by tentacles. The polyp catches small organisms that drift past it in the water current. It can reproduce by budding, pinching off new polyps that either stay attached and form a colony or detach and settle somewhere else. The polyp is the stable, sessile phase of the life cycle. It can persist for a long time in conditions that would be difficult for the free-swimming stage. The second body form is called the medusa. This is what most people recognize as a jellyfish. The medusa is the free-swimming stage. It has a bell-shaped body that pulses rhythmically by contracting and relaxing muscle fibers in the bell wall. This pulsing propels the jellyfish gently through the water while also drawing current toward the tentacles, which capture prey. The medusa is the sexually mature stage. It produces eggs or sperm, which are released into the water. Fertilized eggs develop into tiny free-swimming larvae, called planulae. A planula eventually settles on a surface and becomes a new polyp, completing the cycle. In most hydrozoan species, the medusa stage is brief. The animal grows from a polyp, matures into a medusa over a period of weeks, reproduces, and then dies. The medusa is built for reproduction and dispersal, not for longevity. Its body is not engineered to last indefinitely. Death follows reproduction. This is not a flaw. It is a functional design that has served the group for more than 500 million years. Turritopsis dohrnii follows this same blueprint at the start of its life. It begins as a fertilized egg that develops into a planula larva. The planula is tiny, covered in fine hair-like cilia that it beats against the water to swim. It drifts in the current for a brief period, then settles on a hard surface and begins developing into a polyp colony. The colony grows, branching and spreading along the substrate, and eventually begins to bud off tiny medusae. Those medusae detach and begin their free-swimming lives. Over several weeks, a Turritopsis dohrnii medusa matures. Its bell reaches a maximum diameter of about 4.5 millimeters, approximately the size of the nail on your smallest finger. The bell is nearly transparent, with a bright red stomach, visible at the center. The tentacles, between 80 and 90 of them in a fully grown adult, trail in a ring around the edge of the bell. The animal is delicate, soft, and nearly weightless in the water. Up to this point, nothing unusual has happened. This is just a jellyfish growing the way jellyfish have grown for hundreds of millions of years. And then, under the right circumstances, something happens that has not been observed in any other animal in quite the same way. The jellyfish begins to reverse. It does not grow old and die in the way most medusae do. When faced with enough stress, it does something that violates the assumption most biologists held for a long time about how animal development works. It goes back. Not metaphorically, physically, measurably, verifiably. Its cells change their identities. Its body reorganizes, and it becomes a polyp again, starting the life cycle from a younger stage, carrying the same genetic material, but beginning again with cells that have been reset. That is what the next several parts of tonight's journey will be about. How that happens, what it requires, what it means, and why a creature with no brain, drifting in the ocean without any apparent awareness of its own situation, has evolved what is arguably the most remarkable cellular trick known to biology. There is one more detail worth noting about the life of a normal medusa before we move on. When a medusa dies in the ordinary way, it does not leave a body that persists. Jellyfish bodies are mostly water. They are held in form by the gentle tension of living tissue. And when that tissue fails, the form dissolves. A dead jellyfish disappears into the ocean in hours. There is nothing left to find. No shell, no skeleton, no durable record of its existence. This makes the study of jellyfish longevity difficult in natural settings. You cannot find old specimens the way you can find old trees with their rings, or old clams with their growth lines, or old fish with their ear bones. The evidence of a jellyfish's age is molecular and behavioral, not structural. And it means that understanding what happens to a jellyfish at the end of its life required watching live animals carefully in controlled conditions, which is exactly what the researchers who first documented the life cycle reversal had to do. What they saw, when they kept stressed or aging medusae under careful observation, was that the expected ending did not always arrive. Instead of declining and dissolving, some animals did something unexpected. Something that, at first, the researchers thought might be an artifact of the laboratory conditions. Something they had to watch many times, in many animals, before they were certain of what they were actually seeing. The medusa was not dying. It was becoming something else. Let your body settle a little more deeply into where you are resting. Let the image form quietly in your mind. A small transparent creature, pulsing in dark water, with a secret written in its genes that the rest of the animal kingdom has not found. The process that makes the immortal jellyfish's life cycle reversal possible has a technical name. That name is transdifferentiation. The word is long, and it sounds complicated, but what it describes is actually something you can understand quite clearly with a simple picture. Start with differentiation. That is the process by which cells develop their identities during the growth of any complex organism. Every cell in your body contains the same genome. A liver cell, and a muscle cell, and a neuron all carry exactly the same DNA sequence, the same complete set of genetic instructions. What makes them different is not their DNA. It is which parts of that DNA they use. During development, as cells divide, and the organism grows from a single fertilized egg into a complex body, cells progressively activate certain genes and silence others. A liver cell activates the genes needed for liver functions and silences everything else. A muscle cell does the same for its own set of functions. A neuron does the same for its own. This activation and silencing is maintained through multiple overlapping systems. There are proteins called transcription factors that bind to specific regions of the DNA and either activate or suppress the genes nearby. There are chemical marks on the packaging proteins around the DNA that make certain regions physically accessible and others physically closed off. There are modifications directly on the DNA molecule itself that influence which genes are read and which are ignored. All of these systems work together to create a stable and consistent cell identity that is maintained reliably through thousands of cell divisions. The stability is essential. Your liver has been making liver cells for your entire life. It does not accidentally make muscle cells or skin cells. The identity is locked in, and for the coordinated function of a complex organism, that lock needs to be reliable. Transdifferentiation is what happens when the lock is opened. In transdifferentiation, a cell that has already committed to one identity changes into a different identity. The transcription factors that defined it as one cell type are displaced. The epigenetic marks that enforced its identity are erased or rewritten. The molecular memory of what it was is cleared, and new instructions are written in its place. The cell becomes something it was not before. In most animals, this is extremely rare and usually happens only in pathological circumstances, such as when cancer cells acquire the ability to invade new tissue types by changing their surface characteristics. In most normal biology, the differentiation of cells is treated as a one-way process. Cells specialize. They do not unspecialize. In Turritopsis dohrnii, transdifferentiation happens at the scale of the entire organism. When the jellyfish begins to reverse its life cycle, the cells of the adult medusa begin to lose their specialized identities simultaneously. The muscle cells that powered the pulsing of the bell, the cells of the digestive system, the cells of the reproductive organs, the structural cells of the tentacles, all of them begin to withdraw from their committed identities. The molecular marks that defined each cell type are cleared. The cells enter a state of reduced differentiation, a state closer to what embryonic cells look like before they have committed to a specific fate. And then, from that less differentiated state, they begin to rebuild. They receive new instructions. They redifferentiate into the cell types required for a polyp, which is a completely different body organization with different structures and different functional requirements. A cell that was contracting to drive the jellyfish's swimming motion becomes part of the stationary feeding apparatus of a polyp. A cell that was part of the reproductive system of the adult medusa becomes part of the attachment structure anchoring the new polyp to the seafloor. The transformation is total. The entire cellular inventory of the organism is reorganized. This was first documented by researchers including Stefano Pirano and Christian Sommer in the 1980s and 1990s, working with specimens in the Mediterranean Sea. Their initial observations were met with skepticism because what they described seemed to contradict one of the established principles of animal development. They spent years confirming and reconfirming their results before the broader scientific community accepted what they were seeing. What makes the jellyfish's transdifferentiation particularly remarkable from a biomedical perspective is not just that it happens, but that it happens safely. In laboratory experiments with mammalian cells, when scientists try to force cellular reprogramming using introduced proteins, the process frequently produces cells with unstable genomes or abnormal behavior. Forced reprogramming in mammals carries a risk of triggering cancer, because cells that lose their committed identity can sometimes begin proliferating without the normal constraints. The immortal jellyfish reprograms the identity of every cell in its body, safely, repeatedly, in under 2 days, with no apparent genomic instability and no uncontrolled growth. How it achieves this precision is one of the key open questions in the field. The most current research, published in journals including G3 Genes Genomes Genetics, and in the 2022 Genome Comparison Study from the Barcelona Institute of Marine Sciences, has begun to map the molecular systems involved. And the picture that is emerging is one of a very simple animal with remarkably sophisticated cellular machinery for maintaining genomic integrity while simultaneously erasing and rewriting cellular identity. Think for a moment about what this means at a practical level. Your cells right now know what they are. They are confidently specialized. They are carrying out their specific functions with precision. That confidence, that specificity, is what allows the coordinated function of your body. And the immortal jellyfish can, in the right circumstances, take every cell in its body and temporarily dissolve that confidence entirely, then rebuild it in a new configuration. It is not magic. It is chemistry. It is gene regulation. It is the same molecular language that every living cell uses, applied in an arrangement that most organisms do not carry. Let your thoughts drift gently around that. A cell forgetting what it is, briefly becoming undefined, and then reading new instructions about what to become next. In a creature with no brain, in the dark water, without any deliberate intention behind it at all. Between what the jellyfish was and what it will become, there is a third state. It is called the cyst stage, and it is one of the most biologically unusual structures documented in the animal kingdom. The cyst is where the molecular work of the life cycle reversal actually happens. It is the threshold, the moment of transition, the state of maximum cellular uncertainty, where the body is neither medusa nor polyp, but something briefly in between. When a stressed medusa begins to reverse, the first visible changes are the retraction of the tentacles and the contraction of the bell. The swimming stops, the animal sinks. The organized form of the adult medusa, its layers of tissue, its internal organs, its clear structural definition, begins to fold inward and compress. Over several hours, the entire body collapses into a small, dense mass of tissue that settles on the seafloor. Under a microscope, the cyst looks like a cluster of cells with very little of the organized structure visible in either the medusa or the polyp. The boundaries between different tissue types, which are crisp and well-defined in the adult, have blurred. The cells have lost the visual characteristics of their previous specialized identities. The cyst is round, compact, and apparently formless. But the cyst is not inactive. This is a critical point. The cyst is not dormant. It is not like a seed or a spore, simply waiting for better conditions in a metabolically quiet state. It is intensely active at the molecular level. The genes are being transcribed. The proteins are being produced. A complex and coordinated program of cellular reprogramming is underway. The cyst only looks quiet from the outside. Research teams studying the transcriptome of the cyst, which means measuring which genes are being actively read and turned into protein at any given moment, have revealed a specific and striking pattern. The study by Christine Schnitzler and colleagues at the University of Florida, published in the journal G3 in 2019, was one of the first systematic characterizations of this. In the cyst, genes associated with cellular differentiation are suppressed. The molecular programs that define specialized cell identities have been turned down or turned off entirely. At the same time, several other categories of genes are highly elevated compared to both the medusa and the polyp stages. The most elevated categories include genes associated with DNA repair. Multiple pathways for detecting and correcting DNA damage are running at higher levels in the cyst than in any other stage. Genes involved in repairing double-strand breaks, which are among the most serious forms of DNA damage, are particularly active. The cyst is, in essence, using this period of cellular reorganization to perform a comprehensive check and repair of the genome. Telomere maintenance genes are also elevated. Telomerase, the enzyme that extends telomere length, is more active in the cyst than in the mature medusa. This suggests that during the cyst stage, the shortening of telomeres that accumulated during the medusa's life is being partially or fully reversed. The cellular clock, in this specific and measurable sense, is being reset. Genes associated with pluripotency are also upregulated. Pluripotency refers to the state of a cell that has not yet committed to a specific identity and retains the ability to become many different cell types. Embryonic stem cells are pluripotent. Adult specialized cells are not. But during the cyst stage, the cells of the jellyfish express genes that resemble the pluripotency-maintaining genes of embryonic cells. They become, briefly, something closer to stem cells. Open to new instructions. Capable of a different future. The genome assembly published by Shin Kubota's group at Kyoto University in 2022, in the journal DNA Research, identified 23,314 genes in the Turritopsis dohrnii genome. Comparison of expression patterns across the polyp, medusa, and cyst stages revealed clear differences consistent with the transcriptome studies. The cyst has a distinct molecular identity, not a mixture of medusa and polyp patterns, but something genuinely different. It is a specialized state for doing a specific job, which is the safe and comprehensive reorganization of an entire organism's cellular identity. The duration of the cyst stage is brief given the complexity of what it accomplishes. Under laboratory conditions, the entire transformation from mature medusa to new polyp takes approximately 24 to 36 hours. Within that window, telomeres are lengthened, DNA is repaired, differentiation marks are erased, new differentiation marks appropriate for a polyp are laid down, and the physical structure of the new polyp begins to emerge. The cyst is perhaps the most alive moment in the jellyfish's life cycle. It is the moment when nothing is fixed and everything is being rewritten, when the boundaries between what was and what will be are dissolved and rebuilt. And it happens on the floor of the sea, in the dark, in a mass of cells so small, it is nearly invisible, in an animal with no brain and no awareness of what it is doing, simply following instructions written into its genome hundreds of millions of years ago. There is also a timing dimension to the cyst stage that is worth appreciating. The transformation from fully mature medusa to new polyp takes approximately 24 to 36 hours under laboratory conditions. That is less than 2 days. In that window, the jellyfish completes what is arguably the most complex biological transformation known in any animal. Every cell in the organism changes its molecular identity. Telomeres are restored. DNA is repaired across the entire genome. Epigenetic marks are erased and rewritten. The physical structure of an entirely different body form emerges from the reorganized cells of the previous one. Compare this to what happens when scientists try to reprogram mammalian cells in a laboratory. The process of generating induced pluripotent stem cells from adult human cells typically takes 2 to 4 weeks, requires sustained exposure to reprogramming factors introduced artificially, and frequently produces cells with genomic instability. Scaling this process to reprogram an entire organism all at once, safely, in under 2 days, is something no laboratory technology has achieved. The jellyfish does it every time it is stressed enough to trigger the reversal. Without any equipment, without any externally introduced factors, using only the molecular machinery encoded in its own genome, activated by signals arising from within its own cells. The efficiency of this process is one of the reasons researchers find the cyst stage so compelling as a subject of study. Understanding how so much reorganization happens so quickly and safely would be a significant advance, not just in the biology of this one species, but in the broader understanding of what cellular reprogramming can accomplish. Let the image rest with you quietly. A threshold state. A moment between one version of a life and the next. The biology of beginning again. For most of the decades since the immortal jellyfish's ability was first observed, scientists could watch the transformation happen, but could not see the molecular machinery that drove it. They could document the phenomenon. They could describe it. But they could not yet read the instructions. That changed with advances in genomic sequencing. In 2022, a research team led by Maria Pascual-Torner at the Institute of Marine Sciences in Barcelona published a detailed comparison of the genome of Turritopsis dohrnii with the genome of Turritopsis rubra, a closely related species that does not undergo life cycle reversal. This study was published in the Proceedings of the National Academy of Sciences, one of the leading scientific journals in the world. By placing the two genomes side by side and identifying the differences, the team could begin to answer a question that had been central to the field for decades. What, at the level of DNA, is actually different about the immortal jellyfish? What they found was a picture involving approximately 1,000 genes related to aging and cellular maintenance, with differences scattered across multiple biological systems rather than concentrated in any single pathway. The first major finding was in DNA replication and repair. Turritopsis dohrnii carries additional copies of genes encoding DNA polymerases. These are the enzymes that perform the actual work of copying the DNA sequence when a cell divides. Having more copies of these genes is associated with greater replicative fidelity, a higher probability that each copy of the DNA sequence is made accurately and completely. Beyond that, the genome shows duplications and enhanced variants in multiple DNA repair pathways, including pathways for repairing double-strand breaks in the DNA molecule. Double-strand breaks are one of the most dangerous forms of DNA damage because they sever the chromosome entirely and must be precisely repaired, or they can lead to chromosomal rearrangements. Enhanced capacity for this kind of repair means that errors accumulate more slowly over time, and the cellular aging that follows from accumulated DNA damage proceeds more slowly, or, in the context of the cis stage, can be more fully reversed. The second major finding involved oxidative stress response. All cells that use oxygen to generate energy produce reactive oxygen species as a byproduct. These reactive molecules are chemically aggressive. They can damage DNA, they can damage proteins, and they can damage the lipid membranes that surround cells and their internal organelles. The damage they cause accumulates over time and contributes to aging. Every organism has antioxidant systems, molecular defenses that neutralize reactive oxygen species before they can cause harm. But those systems are never perfectly efficient. In Turritopsis dohrnii, the genome shows amplification of several genes associated with the response to oxidative stress. More copies of these genes means a larger pool of the protective proteins they encode. The jellyfish has, in effect, a more powerful antioxidant defense system than its mortal relative. Reactive oxygen species are neutralized more efficiently. DNA and protein damage from oxidation accumulates more slowly. The third finding was in telomere maintenance. The genome comparison identified specific gene variants and gene copy number differences in systems related to telomere length and integrity. This was consistent with the earlier transcriptome studies showing elevated telomerase activity in the cis stage. The capacity for telomere maintenance in this species appears to be built into the genome architecture rather than being only an emergency response. The tools for preserving telomere length are more abundant and apparently more effective than in related species. The fourth finding connected to the molecular machinery of cellular reprogramming itself. The study identified gene expression patterns that show similarity to the Yamanaka factors, a set of 4 proteins first identified by Shinya Yamanaka in 2006, for which he was awarded the Nobel Prize in Physiology or Medicine. The Yamanaka factors, which include proteins known by the abbreviations OCT4, SOX2, CALF4, and c-MYC, are able to reprogram differentiated adult mammalian cells back into an embryonic-like state when they are experimentally introduced into those cells. The immortal jellyfish appears to have functional analogs or related systems that activate during the life cycle reversal, providing a native reprogramming capacity that most organisms simply do not carry. What is important to understand about these findings is not just that they confirm what scientists already suspected. It is that they show the ability of Turritopsis dohrnii to undergo life cycle reversal is not the product of a single special gene. There is no single immortality gene. The ability arises from the coordinated interaction of many genetic systems, each one enhanced in specific ways relative to related species. Better repair, better protection, better maintenance, better reprogramming. None of these alone would produce the result. Together, they do. This has implications for how researchers think about the biology of aging. Aging is a multi-process phenomenon. And what this animal demonstrates is that resistance to aging and the capacity to reverse it is also a multi-process achievement. There is no shortcut. There is no single target. There is a system, and the system works when all its components are well configured. The 2022 Barcelona study also identified something important about the distribution of these genetic differences. They are not concentrated in a single biological system or a single region of the genome. They are spread across multiple systems and multiple chromosomes. This scattered distribution suggests that the ability of Turritopsis dohrnii to perform life cycle reversal is not the product of a single evolutionary event, a single mutation, or a single gene duplication that changed everything. It is the cumulative result of many separate improvements, accumulating over a very long evolutionary history, each one contributing a small increment to the overall capacity. This has implications for how researchers think about what evolution can produce when selection pressure operates consistently in a particular direction over a very long time. The immortal jellyfish lineage has presumably been experiencing selection pressure related to survival under stress for hundreds of millions of years. The result is a genome that has been refined and tuned across an immense stretch of time, with enhancement after enhancement adding to a system that has grown progressively more capable of resisting and reversing cellular aging. We are looking at the product of evolutionary refinement operating at a timescale we cannot easily imagine, and what that refinement produced, in this particular lineage, in this particular ocean, is the most effective cellular rejuvenation system known to science. Rest with that idea for a moment. 1,000 genes, working together, accumulated by evolution over hundreds of millions of years, producing something that the rest of the animal kingdom has not independently arrived at in quite the same way. Turritopsis dohrnii is not the only living thing that has pushed against the rules that govern most of biology. In the wider world, across many different branches of the tree of life, other organisms have found their own ways to resist aging, to slow it, to nearly escape it, or in some cases to approach something that functionally resembles negligible senescence, which is the technical term for an organism that does not show measurably increasing mortality with age. Understanding these other cases helps us see the immortal jellyfish not as a singular freak of nature, but as the most extreme point on a spectrum that biology has been exploring in many places at once. Start with the hydra. The hydra is a small freshwater animal, a few millimeters long, related to jellyfish within the cnidarian group. It lives in ponds and slow-moving streams, attached loosely to surfaces by its foot, with a mouth at the other end surrounded by tentacles it uses to catch small prey. Hydras have been studied extensively in aging research because they appear to show negligible senescence in laboratory conditions. Population studies maintained over multiple years show no measurable increase in death rate with age, no decline in reproductive output, no characteristic signs of aging accumulation. The biological basis for this appears to be related to the hydra's stem cell population. Roughly 1/3 of all cells in a hydra at any given moment are stem cells. These stem cells continuously replace all other cell types throughout the animal's body. Differentiated cells that accumulate damage are replaced by fresh cells, differentiating from the stem pool. The stem cells themselves maintain extraordinarily high telomerase activity, keeping their telomeres long and their replicative potential intact. The hydra essentially renews its cellular population on an ongoing basis, preventing the accumulation of damaged cells that drives aging in most organisms. The ocean quahog is a clam found in the cold waters of the North Atlantic. Individual specimens have been dated at over 500 years of age using the growth rings on their shells, which accumulate annually and can be counted the way tree rings are counted. The oldest known individual, nicknamed Ming, was estimated at 507 years when it was discovered. These clams do not show the typical pattern of cellular aging within their normal lifespan. Their cells maintain high antioxidant capacity and genomic stability over centuries. Their longevity is thought to be related to extremely efficient management of oxidative damage and a particularly slow metabolic rate. The naked mole rat is a small burrowing rodent found in the arid regions of East Africa. It is remarkable among mammals for several reasons. Its maximum recorded lifespan is over 37 years, which is roughly 10 times longer than would be predicted based on its body size. Small rodents generally age quickly because their high metabolic rates produce more oxidative stress per unit of time. But the naked mole rat appears to manage this with unusual efficiency. Its cells show enhanced protein quality control, meaning they more effectively identify and remove damaged or misfolded proteins. Its cancer incidence is remarkably low despite its long lifespan, and its death rate does not increase substantially with age until very late in its life, placing it in the category of negligible senescence among mammals. Greenland sharks are among the longest-lived vertebrates known to science. They inhabit the cold, deep waters of the North Atlantic and Arctic Oceans. Radiocarbon dating of crystalline proteins in their eye lenses, proteins that are laid down at birth and never replaced, has estimated the ages of individual sharks at over 400 years. The oldest specimen in that study was estimated to be around 392 years old, with an uncertainty range that extended to over 500 years. They grow extremely slowly and do not reach sexual maturity until they are approximately 150 years old. Their extreme longevity is thought to be partly a consequence of their cold environment, which slows metabolic rate and therefore slows the accumulation of oxidative damage, and partly of biological adaptations not yet fully characterized. Lobsters are often cited as examples of negligible senescence, and while this is somewhat overstated, they are genuinely interesting in the context of aging. Lobsters express high levels of telomerase in nearly all of their tissues. Unlike most animals where telomerase is restricted to specific cell types like stem cells, this means their telomeres do not shorten significantly with age, removing one of the standard mechanisms of cellular aging. Older lobsters do not show the characteristic increase in mortality rate with age that most animals show. However, they do eventually die, often from the increasing energetic cost of molting their shells as they grow, rather than from the kind of cellular deterioration that kills most organisms. What all of these diverse examples share is a common theme. Organisms that resist aging have generally evolved enhanced versions of one or more of the following capabilities: better DNA repair, better management of oxidative stress, better protein quality control, better telomere maintenance, more active stem cell renewal, slower metabolic rates. They have not escaped the fundamental mechanisms of cellular damage. They have evolved to resist those mechanisms more effectively than most. Turritopsis dohrnii is different from all of these in a specific and important way. The others slow aging or reduce its effects. The immortal jellyfish reverses aging that has already occurred. It does not just resist the accumulation of cellular damage. When the damage accumulates past a threshold, it dismantles the damaged system entirely and rebuilds it fresh from the same materials. That distinction between slowing and reversing is what sets it apart. Not just in degree, but in kind. It is not doing what the others do better. It is doing something qualitatively different, and that is why it continues to attract serious scientific attention decades after its ability was first described. Science is not a collection of finished answers. It is a process of refining questions. And the immortal jellyfish is a subject that has generated better and better questions over the 4 decades since its ability was first observed. Let us be clear about what the science currently knows, and equally clear about what it does not yet know. What we know with high confidence is this: Turritopsis dohrnii genuinely undergoes life cycle reversal. The transformation from mature medusa to new polyp has been documented in laboratory settings by multiple independent research groups in multiple countries. It has been filmed. It has been documented in detail using microscopy. It has been induced artificially by researchers using mechanical damage, salt concentration changes, heat shock, and chemical treatment with cesium chloride. The phenomenon is not in question. It is real. The transformation involves genuine cellular transdifferentiation. The cells of the adult medusa change their identities and become the cell types of the polyp. This is not reproduction. The new polyp is not a genetically distinct offspring formed from eggs or sperm. It is built from the reorganized cells of the original adult. The 1996 paper by Pirano and colleagues documented that even portions of the medusa that lack the interstitial stem cells that would normally be responsible for regeneration are still capable of full life cycle reversal. This confirmed that the transformation involves transdifferentiation of differentiated somatic cells rather than proliferation from a pre-existing stem pool. The cis stage has a distinct molecular identity. The transcriptome studies from 2019 and the genome assembly from 2022 both confirm that the cyst is a specialized state characterized by suppressed differentiation pathways and elevated DNA repair, telomere maintenance, and pluripotency-related gene expression. The molecular pattern is consistent with a genuine cellular reprogramming event, not simply a stress response or a dormancy. The genome of Turritopsis dohrnii shows specific genetic differences from its mortal relative that are consistent with enhanced capacity for DNA repair, oxidative stress resistance, telomere maintenance, and cellular reprogramming. This was established by the 2022 Barcelona study. What we do not yet know with the same confidence is the precise mechanistic sequence. We do not have a complete step-by-step account of what triggers the reversal. When a medusa experiences stress, what exactly is the first molecular signal that initiates the process? There is evidence suggesting a role for the Dublin signaling pathway, a well-conserved developmental signaling system used throughout the animal kingdom. One study found that a dobuint-related gene is expressed at 10 times its normal level in medusae undergoing reversal compared to unstressed adults. But the full cascade of signals, from initial stress perception to cyst formation to polyp reconstruction, is not yet mapped. We do not fully understand the epigenetic dimension. Epigenetic marks, meaning chemical modifications to the histone proteins that package DNA and to the DNA molecule itself, are central to cellular identity. Erasing and rewriting these marks is a necessary part of transdifferentiation. An ongoing research project at the University of Bristol, funded specifically to address this gap, is working to characterize the epigenetic changes that occur at each stage of the reversal. The results of this work will add a critical dimension to the current picture. We do not fully understand how the jellyfish prevents the reprogramming from becoming tumorigenic. In mammalian systems, forced dedifferentiation carries a risk of triggering cancer. The jellyfish reprograms safely. The molecular safeguards that allow this are not yet fully characterized. The research community working on this organism is relatively small. Working with live specimens is technically demanding. The animals are fragile and require carefully controlled conditions. The most dedicated long-term captive population has been maintained by Shin Kubota at Kyoto University, who has observed individual animals undergo life cycle reversal as many as 11 times over a 2-year period under laboratory conditions. The difficulty of working with them means that progress is measured in years and decades rather than months. This is not a limitation of science. It is a reflection of the genuine difficulty of studying a small, delicate marine animal at the molecular level. The tools available today are far more powerful than those available when the phenomenon was first described. Progress is real and ongoing. The gaps in understanding are becoming smaller. What is clear right now is that the ability of this animal to reverse its life cycle is real. It is biological. It follows from specific genetic and molecular characteristics, and understanding it more fully will require continued careful work. It is also worth understanding the pace at which this particular field advances, and why. Working with live Turritopsis dohrnii is technically demanding in ways that working with common laboratory organisms like mice or fruit flies is not. The animals are fragile. They require very specific water conditions, specific temperature ranges, and specific feeding regimes to remain healthy enough to exhibit the behavior being studied. They are small enough that extracting genomic material in sufficient quantities for analysis required, in one key study, collecting DNA from 1,500 individual animals derived from a single clone. The process of establishing a stable breeding colony for research purposes took years of dedicated effort. This means that progress in this field is slower than in fields where the model organism is easier to work with. A research group studying fruit fly aging can run thousands of experiments in the time it takes a Turritopsis dohrnii researcher to establish a healthy colony and observe a single life cycle reversal. The science is not moving slowly because the questions are not important. It is moving at the pace the biology permits. What this also means is that each major result carries particular weight because each one represents a substantial investment of time and resources. The transcriptome studies, the genome assembly, the comparative genomic study, these are not casual observations. They are the products of years of careful work by dedicated researchers who have chosen to invest their careers in understanding one of the most unusual animals on Earth. The patience required by this work is not so different from the patience of the jellyfish itself, which drifts and pulses and waits and, when the time comes, begins again. That is where science stands. Not at the end of this story, but well along in it. The immortal jellyfish did not evolve for our benefit. It did not develop its biology in order to provide medical researchers with a model system. It evolved over hundreds of millions of years in an ocean environment in response to pressures that had nothing to do with human health. And yet, what it has evolved speaks directly to questions that human biology has been wrestling with for as long as there have been biologists to ask them. What causes cells to age? Can that process be slowed? Can any aspect of it be reversed? What are the specific molecular events that determine whether a cell continues to function well or begins to decline? The connections between the jellyfish's biology and human aging research run through several parallel topics. The most direct connection is to telomere biology. In human cells, telomere shortening is associated with cellular senescence and is linked to multiple conditions associated with aging, including cardiovascular disease, certain lung conditions, and some forms of cancer. There is active research into the possibility of using telomerase, the enzyme that extends telomeres, as a therapeutic tool in aging-related conditions. The challenge is doing this safely, because inappropriately activating telomerase can also support cancer growth by giving cancer cells the ability to replicate indefinitely. The immortal jellyfish activates telomere maintenance during the cis stage as part of a broader reprogramming program that includes simultaneous DNA repair and genomic stabilization. How it coordinates telomere extension with these other processes safely is directly relevant to the question of how telomerase-based therapies might be applied safely in human medicine. The second connection is to cellular reprogramming technology. In 2006, Shinya Yamanaka demonstrated that differentiated adult cells from mice and humans could be reprogrammed into induced pluripotent stem cells by introducing 4 specific transcription factors. This was a landmark discovery with profound implications for regenerative medicine. Induced pluripotent stem cells can theoretically be used to generate any cell type in the body, and they can be derived from a patient's own cells, eliminating the problem of immune rejection that complicates transplants. But the current reprogramming process is slow, inefficient, and associated with risks of genomic instability and tumor formation. The jellyfish achieves what is functionally a more extensive version of this reprogramming, safely, in under 2 days, repeatedly. Understanding how it maintains genomic integrity during such a rapid and complete cellular identity change could inform the development of safer and more efficient reprogramming strategies for use in regenerative medicine. This is not a hypothetical connection. Research groups working on the biology of Turritopsis, dohrnii, have explicitly stated that this is one of the primary medical implications of their work. The third connection is to the biology of senescent cells. The accumulation of senescent cells, cells that have stopped dividing and are releasing inflammatory signals, is one of the most active targets in current aging research. A field called senolytic therapy is focused on developing drugs that selectively clear senescent cells from tissues, with the goal of improving tissue function and possibly reducing some of the functional decline associated with aging. Early clinical trials of senolytic drugs have shown promising results in specific conditions. The immortal jellyfish periodically resets its cellular age before senescent cells can accumulate to problematic levels. Understanding the molecular basis for how it does this could provide insights into the design of more effective senolytic or senescence prevention strategies. It is important to be precise about what the research has and has not established in terms of medical application. No current research group has proposed that the biology of Turritopsis dohrnii will produce a human immortality treatment. That is not what the science is suggesting, and it is not what the researchers working on this organism are claiming. What they are saying is more measured. Christine Schnitzler at the University of Florida has described the jellyfish as a model for studying the molecular mechanisms of cellular reprogramming. Maria Miglietta at Texas A&M has framed the scientific significance in terms of what it reveals about aging biology and regenerative potential. These are substantial scientific contributions without requiring any exaggerated claims. The distance between a jellyfish and a human is enormous. We are vastly more complex organisms. We have immune systems, central nervous systems, and somatic differentiation of a kind that has no parallel in any cnidarian. The reprogramming mechanisms that work safely in a 4.5 millimeter animal with no brain cannot simply be transplanted into the context of a human body without enormous additional work. Any application to human medicine would require solving many additional problems that are not present in the jellyfish system. There is a concept in the history of science called proof of principle. A proof of principle is not a complete solution. It is a demonstration that the thing you are trying to accomplish is achievable in principle, under some conditions, in some system. Once you have a proof of principle, the question changes from whether the thing is possible to how to make it work in the context you actually need. Before the immortal jellyfish, the question of whether cellular aging could be reversed was genuinely open. There were reasons to doubt it. The mechanisms of aging seemed deeply embedded in the fundamental biology of cells. The directionality of differentiation seemed fixed. The shortening of telomeres seemed like a ratchet that could only turn one way. Turritopsis dohrnii is a proof of principle that none of those things are absolute. Cellular aging can be reversed. Differentiation can be undone and redone. Telomeres can be restored. The system can be reset. That proof of principle exists now. In a real animal, in a real ocean, in biology that is measurable and reproducible, and documented in peer-reviewed literature. The field of aging research can now ask its questions differently than it could before. That is what the jellyfish has contributed to human medicine. Not a treatment, not an answer. A demonstration that the answer is out there to be found. But the jellyfish has demonstrated something that matters deeply to medicine, regardless of these complications. It has demonstrated that the cellular machinery of aging can be run in reverse under the right conditions. Not gradually slowed, actually reversed. That demonstration changes the conceptual landscape of aging research. It expands the space of what researchers allow themselves to imagine as theoretically possible, and expanding that space, even in ways that do not immediately translate into treatments, is how science advances. There is a philosophical dimension to the story of Turritopsis dohrnii that deserves its own space tonight, because the idea of immortality is one that human minds have grappled with for as long as there have been human minds. It appears in the oldest stories from every culture. It appears in modern fiction and film. And it appears in scientific discussions with a frequency that sometimes outpaces what the evidence actually supports. So let us think carefully about what immortality would and would not mean, even in the most generous biological sense. And let us use the jellyfish as a realistic starting point rather than a dramatic one. The first thing to be clear about is that Turritopsis dohrnii is not immortal in the way the word is usually understood. It is not an animal that cannot die. In its natural environment, most individual Turritopsis dohrnii medusae very likely die without ever undergoing a life cycle reversal. They are eaten by fish, by sea turtles, by other predatory invertebrates. They are killed by disease. They are destroyed by physical events in the ocean. The reversal is available to them as a response to certain kinds of internal stress, particularly stress from damage, starvation, or cellular aging. But it does not protect them from being consumed. It does not protect them from bacterial infection. It does not protect them from physical destruction. What life cycle reversal eliminates is one specific pathway to death, which is the progressive decline and failure of the body's own cellular machinery. That is a significant thing to eliminate. But it is one pathway among several. A Turritopsis dohrnii that lived in a perfectly controlled environment with no predators, no disease, and reliable nutrition might theoretically persist indefinitely through repeated life cycle reversals. But that environment does not exist in nature. The second thing worth considering is the question of identity. When a medusa undergoes life cycle reversal and becomes a polyp, which then eventually buds off new medusae, are those new medusae the same individual as the original? They carry the same DNA sequence. The physical continuity of the cells is maintained throughout the transformation, but the cellular history of the original medusa, the pattern of accumulated differentiation marks, the specific organization of tissue types, the record of what the cells had been, is erased and replaced. There is a classic philosophical puzzle called the ship of Theseus. The question asks whether a ship that has had every single component replaced, one plank at a time over many years, is still the same ship. In the case of the jellyfish, it is more radical than that. The components are not replaced. They are transformed. The same molecules are still there, rearranged into a new configuration, running a new program. For the jellyfish, this philosophical question likely has no experiential dimension. There is no evidence that Turritopsis dohrnii has anything like a persistent sense of self. Its nervous system is a simple nerve net without the capacity for the kind of complex information processing that would be required for anything resembling continuous consciousness or memory. The new medusa that emerges from the life cycle reversal does not remember being the old medusa, because there was nothing in the old medusa that stored or maintained that kind of record. But the question becomes genuinely interesting when we ask what biological immortality would mean for a more complex organism, one with a central nervous system, with memory, with accumulated experience stored in the specific patterns of neural connectivity in the brain. For such an organism, a cellular reset of the kind the jellyfish performs would not simply restart the life cycle. It would erase the record of who the organism had become. The biological continuity would be maintained. The experiential continuity would be destroyed. This suggests that the value of the immortal jellyfish's biology to human longevity research lies not in the concept of a full life cycle reversal applied wholesale, but in the specific molecular components that enable cellular rejuvenation at a more targeted level. Not resetting the whole system, but repairing and maintaining the components that accumulate damage, while preserving the organization of the systems that carry information, memory, and accumulated experience. The third dimension of the paradox is ecological. A species whose individuals can potentially reproduce indefinitely has a very different population dynamic than one whose individuals die after a fixed lifespan. The normal pattern of aging and death serves an ecological function. It turns over generations. It makes room for new individuals carrying new genetic combinations. It allows populations to adapt as environments change. An organism that never ages and can repeatedly generate new offspring from its own reorganized cells has, in principle, an unlimited capacity for population expansion from a small number of founders. Maria Miglietta has noted in her research that a single jellyfish that does not die can produce hundreds of new jellyfish introduced into the environment. This is partly why Turritopsis dohrnii has been so successful as an invasive species. Carried around the world in the ballast water of cargo ships, small populations in new ocean environments can potentially expand rapidly because even a single individual, if it undergoes life cycle reversal, produces a new polyp colony that generates new medusae. The population does not need to maintain minimum numbers the way sexually reproducing populations with fixed lifespans do. Immortality is not a simple gift. It is a trade. And what is given up in that trade is generation turnover, the evolutionary mechanism by which populations adapt. Let those ideas settle around you slowly. The jellyfish is not trying to teach us anything. It is not a message or a symbol. It is a biological system doing what its genes instruct it to do. The meanings we find in it are meanings we bring. But they are not empty meanings. There is something genuinely worth thinking about in what this animal reveals about the relationship between life and time. We have come a long way tonight. We began with the biology of aging. We followed the story of how cells accumulate damage over time, how telomeres shorten with each division, how epigenetic marks drift and lose precision, how mitochondria deteriorate and proteins aggregate, and how the sum of all these processes eventually overwhelms the body's ability to compensate. We understood why evolution has not produced a universal solution to aging, because selection favors reproduction over longevity. Once reproduction has been achieved, we traveled into the ocean and followed a small transparent jellyfish through its life cycle. We watched it grow from a drifting larva to a settled polyp to a free-swimming, sexually mature medusa, pulsing quietly through warm water with its bright red stomach and its ring of fine white tentacles. We watched it, under enough stress, begin to reverse. We saw the tentacles retract. We saw the bell contract. We watched the organized form of the adult dissolve into a small cluster of cells on the seafloor. And we understood that this was not death. It was the beginning of a transformation. We explored transdifferentiation, the process by which fully committed adult cells dissolve their specialized identities and rebuild themselves into something new. We understood why this is so unusual in biology, why the stability of cell identity is essential for complex organisms, and why the jellyfish's ability to perform this transformation safely and completely in under 2 days is something that laboratory science has not yet replicated in any other system. We spent time in the cis stage, that threshold state between one life and the next, where DNA repair runs at its highest levels, where telomere maintenance enzymes work to restore the cellular clock, where pluripotency genes activate and the cells become briefly open to new instructions. We saw the molecular identity of the cyst in the transcriptome data and understood that it is not a passive state, but an intensely active one. We looked at the genome comparison that identified approximately 1,000 gene differences between Turritopsis dohrnii and its mortal relative, covering DNA repair, oxidative stress response, telomere maintenance, and cellular reprogramming capacity. We understood that the immortal jellyfish's ability does not arise from a single special gene, but from the coordinated interaction of many enhanced systems working together. We placed the jellyfish in the broader context of other long-lived and negligibly senescent organisms, from the hydra to the ocean quahog, to the naked mole rat to the Greenland shark. And we understood that what the jellyfish does is qualitatively different from what any of the others do, not slowing aging but reversing it. We examined what the science knows with confidence and what remains to be discovered. We thought about the implications for human medicine, for telomere biology, for cellular reprogramming technology, for the field of senolytic therapy, and we were honest about the enormous distance between a 4.5 millimeter cnidarian and a human being. We sat with the philosophical dimensions of immortality. We questioned what it would mean for a more complex organism. We considered the ecological paradox of unlimited reproduction without death. And we came to understand that immortality, even the partial and conditional immortality of the jellyfish, is not simply the absence of death. It is a different kind of existence, with its own costs and its own ecological implications. And now we arrive at the end of this journey. What does the immortal jellyfish actually teach us? It teaches us that the rules are not as fixed as they seem. Aging appears to be universal. Every complex organism ages. Every body eventually accumulates enough damage that the system fails. This appears to be a biological constant, a feature of life written into the nature of how cells work. But it is not an absolute constant. It is a nearly universal outcome arising from specific molecular mechanisms. And specific molecular mechanisms can, in principle, be arranged differently. Turritopsis dohrnii carries a different arrangement. Not by design, not by intent, by the slow and patient work of evolution over hundreds of millions of years, in the context of an ocean that has always been a laboratory for biological possibility. It teaches us that the capacity for renewal is not a fantasy. It is biology. It is encoded in the genome of a real animal that is alive right now, drifting in real oceans around the world, doing its quiet cellular work without any awareness of the centuries of scientific attention that has been directed toward it. It teaches us that simple organisms can carry molecular sophistication that complex organisms lack, that the tree of life has many branches, and that some of those branches have found solutions to fundamental problems that the vertebrate lineage, for all its complexity, has not found. That paying attention to unfamiliar organisms in unfamiliar environments is one of the most reliable ways to expand the boundaries of what biology thinks is possible. And perhaps most quietly, it offers an image worth carrying into sleep, somewhere in the Mediterranean Sea right now, or in the Pacific, or in the Atlantic, or in the waters off the coast of Japan where Shin Kubota has been caring for a captive population for decades. A small transparent animal is drifting in the dark. It pulses slowly. Its tentacles trail in the current. Its bright red stomach glows faintly through its transparent bell. It has no brain. It does not know it is remarkable. It does not know that scientists have been studying it for 40 years, or that research teams around the world are working to understand what its genome contains, or that its biology has been described in papers and journals and science magazines. And now, tonight, in this quiet voice accompanying you towards sleep, it simply exists. It pulses and drifts and feeds and waits for the moment when the signal comes that says, it is time to begin again. And when that signal comes, it will fold inward, become briefly undefined, and rebuild itself. Quietly. In the dark. Without any hurry. Now you can let all of this settle. Let the ideas become soft and formless, like the cyst itself, suspended between one state and the next. Let your body become heavy. Let your breathing slow to its own easy pace. The science has done its work tonight. It has shown you something real and beautiful and strange. And now it can rest alongside you. The ocean continues. The jellyfish pulses. And somewhere in the dark water, a small cluster of cells is doing something ancient and remarkable, following instructions written long before any of us were here to wonder about them. If this journey moved something in you, you are welcome to like or subscribe, or leave a thought below. It helps more people find this channel. One quiet mind at a time. For now, rest. Sleep well. Good night.